Welcome back, everyone. I'm glad you're back for class. This is cell biology, and we are talking about the properties of cells. We just talked about how cells emerged on our Earth, and now let's kind of explore what those different characteristics of cells are. So, when we're describing cells, we can differentiate them based on their organizational complexity. So, how large they are, how complicated the different components of them are. We can characterize them based on their molecular components, what kinds of molecules are in the different cells, which is different through different cell types, what size they are, what shape they are, that's different in different cell types, and how they're specialized. There are two general types of cells, prokaryotes and eukaryotes. Prokaryotes would be the simpler cells. This is what bacteria and archaea are made of. Bacteria you may know of as being things that cause disease, although many bacteria are important in many ways beyond that. You may not have heard much about archaea, but archaea are also kind of a more simple kind of cell, kind of in many ways similar to bacteria, but also in many ways similar to eukaryotic cells. Eukaryotes are the more complex type of cells, and these are the cells that we see in plants, in animals, in fungi, in algae, in protozoans. Anything that is made of a cell that is not a bacteria or an archaea is probably a eukaryote. The main difference between prokaryotes and eukaryotes is that a eukaryote has their DNA included inside of a membrane-bound nucleus. So that means around the DNA is a membrane, creating a distinct nucleus in that cell. In a prokaryotic cell, there is not a nucleus. They do have DNA in the cell, but the DNA is not enclosed within that membrane. The DNA is found within the cytosol. Besides just saying prokaryotes and eukaryotes, there's really a lot of divergence within those prokaryotes. Bacteria and archaea, although they're both microscopic, it's very easy to just throw them all into that same basket. They're very different. A bacteria and an archaea are just as different from each other as a bacteria and a human are different from each other. So it's not really accurate to just throw cells into prokaryotes and eukaryotes. Instead, we have three domains. Eukaryotes, which are all of the organisms that have eukaryotic cells. Eukaryotes would include plants, animals, fungi, protozoans. And then our other two domains are both prokaryotic. We have our archaea and our bacteria. So eukarya would be our first domain here with fungi, animals, plants, algae. Archaea have halophiles, which would be organisms that live where it's very salty. Thermophiles, which live where it's very hot, thermo like thermometer, and methanobacteria, which are not bacteria, they are archaea. They produce methane, so like found in intestines of ruminants, cows that produce a lot of methane. It's these archaea producing it. Then bacteria would be things like chlamydia, photosynthetic bacteria, E. coli, streptococcus, these kind of things would all be considered different types of bacteria. Okay, so let's compare bacteria, archaea, and eukarya. So, expecting that bacteria and archaea are both prokaryotic, it's not surprising. They are both small. They do not have a nucleus or organelles. Compared to eukaryotes, which are larger, we do have a nucleus, we do have our organelles. Eukaryotes have actin and tubulin proteins. These are the microtubules and microfilaments that help give the cell its shape. It's cytoskeleton. It's the skeleton for the cell. Bacteria and archaea do have proteins that give the cell its shape also, but they're not actin and tubulin. They're actin-like proteins and tubulin-like proteins. Eukaryotes are able to do exocytosis and endocytosis. That's a specific type of process that allows us to bring things into the cell, endo, bringing in, or exo, exiting the cell, taking them out. So this is a specific process that eukaryotes are able to do to bring things in and out of the cell. Bacteria are not able to do that. Then here in our cell walls, we start to see some divergence between bacteria and archaea. Bacteria have a cell wall made of peptidoglycan. This is a sugar protein matrix, could be thin or thick. Archaea tend to have not peptidic glycan, but other proteinaceous types of cell walls. Sometimes pseudomurine is one you'll hear a lot. Then eukaryotes don't always have a cell wall. So animals do not have a cell wall. We don't have a cell wall in our cells. But eukaryotes, when they do have a cell wall, it's going to be typically some sort of sugar type of cell wall. So plants, we see cellulose mainly. Algae, we see cellulose. You could also see pectin. Fungi, mainly we see chitin. You could also see cellulose. Animals and protozoans don't have cell walls. Because bacteria and archaea do not have a nucleus, when their cell divides, there is no process of dividing the nucleus. It's just copy your DNA. Once you have 2 copies of DNA, split your cell in half. That's binary fission, basically. So that's how they divide. Eukaryotes, because we do have a nucleus, we have to first divide the nucleus before we can divide the rest of the cell. So our cell division involves mitosis or meiosis, followed by cytokinesis. So division of the nucleus, followed by division of the cytoplasm and cell stuff. Then when we start looking at nucleotide stuff is where we start seeing archaea diverging from bacteria and seeming more similar to eukaryotes. So bacteria typically have circular DNA and not a lot of proteins around it. Because their DNA is circular and smaller, there's not so much like tangling and messing that they need to organize it quite so carefully, so they don't have as many proteins. Eukaryotes have linear chromosomes. That means we make straight lines, and we usually wrap them up around proteins called histones. So think of like a spool of thread. The histone is the spool, and the chromosome is the thread. So chromosomes are wrapped around these spools of histone. Archaea do usually have circular chromosomes, but they typically are associated with these histone-like proteins. So we are starting to organize that DNA around these histone proteins here. In bacteria, we have minimal RNA processing. In fact, once RNA is being made, it's almost immediately being translated into a protein. In archaea, we see a moderate amount of RNA processing. That means once RNA is made, there's some processing before that RNA is functional. And in eukaryotes, we see a lot of RNA processing. We'll talk about exons and introns and a 5' cap and a poly A tail. There's a lot of processing that's going to happen before that RNA is functional as a molecule. Bacteria have a bacterial type of transcription initiation. That means the way that they start creating RNA is the bacterial type, not surprising. Eukaryotes have a eukaryotic type of transcription. So we've created our RNA slightly differently. And archaea here have a eukaryotic type of transcription initiation. So it's more similar to eukaryotes in how they start creating RNA. Bacteria have a bacterial type RNA polymerase. Eukaryotes have a eukaryotic type RNA polymerase, or polymerases. And archaea have features of both. Ribosomes in bacteria and eukaryotes are different. There's these different numbers that are actually going to be kind of important. Bacteria have a 70S ribosome. Eukaryotes have an 80S ribosome. That means that they're different sizes. Bacteria are a little bit lighter, less dense, and also slightly different shapes. And also a difference in the number of proteins. Although this seems kind of a random fact, this is a really big difference that is used by a lot of antibiotics. So something like streptomycin can attack a bacterial ribosome and kill a bacterial cell without affecting our ribosome. So we are not harmed by taking that medicine. Archaea have a 70S ribosome, similar to bacteria, although they do have more proteins and ribosomal RNAs. So they're kind of a nice in the middle here. Bacteria have a bacterial type initiation of translation or creating proteins. Eukaryotes and archaea have the eukaryotic type. And then we also have differences in our phospholipids making up our membranes. So there's a lot of information here that like one line is not necessarily related to the next line. It's a lot of kind of random stuff. But it also lists the different chapters. We're going to come back and talk about not all of these, but many of these in future chapters. But if you do want to know more about it, you are able to go back and read some of those chapters. Or if you're okay to just kind of memorize this kind of table. Right now, the main thing is to be able to understand that bacteria, archaea, and eukaryotes are different in a lot of different ways. And although bacteria and archaea are both prokaryotes, they are also very different. There's not a lot of consensus yet if bacteria evolved into archaea, which evolved into eukaryotes, or if bacteria is split, or if archaea is split. There's a lot of conflicting evidence that we haven't yet quite worked through to really know which domain came first and how that led to all of the other ones. So I'm not implying any sort of ancestry in these tables either. It's just comparing those different traits of these different cells. Okay, there are some shared characteristics between these three domains, between eukarya, archaea, and bacteria, but each one of these do have many distinguishing properties. Eukaryotes have a true membrane-bound nucleus. That is one you actually need to know all the way. Eukaryotic cells have a true membrane-bound nucleus. Prokaryotes do not. However, that does not mean that they do not have DNA. They do still have DNA, but that DNA is going to be folded up and wrapped up into a structure called the nucleoid. The nucleoid would be this purple area here. It is the DNA floating in the cytosol. Or here you can see the nucleoid floating in the cytosol. It's not just free to move around wherever it feels like, though. It is attached to the membrane, and there will be specific areas of that cell where the nucleoid is going to be remaining. Bacterial and archaeal cells do not usually contain internal membranes at all. So not only do they not have a membrane around their nucleus, they also don't typically have a membrane around organelles like a mitochondria or a Golgi apparatus. One exception here, though, is that there are some photosynthetic bacteria called cyanobacteria, and they do have a lot of internal membranes, a lot of extensive folded membranes inside of their cells, where a lot of photosynthetic enzymes are organized. And this is where a lot of photosynthetic reactions are carried out. Cyanobacteria are really important in photosynthesis, creating oxygen for our planet. And some bacteria do have some membrane-bound or protein-lined structures that kind of resemble organelles. All eukaryotes, in contrast, have extensive organelles. We have a lot of internal structuring and compartmentalizing. So you can think of like the prokaryotes as a little mom and pop shop where they might have one or two employees who do all of the work. Eukaryotes are like the big mega store where you have a separate department for, rotating your tires, and a separate department for selling diapers, and a separate department for selling toothpaste. And those different departments have specific tasks that the employees do in them. Within our eukaryotic cells, those different compartments are called organelles, and they include things like the endoplasmic reticulum. This is an animal cell. Here's the endoplasmic reticulum, or an endoplasmic reticulum here in a plant cell. A Golgi complex here in our animal cell. Mitochondria. Mitochondria are where we produce energy, like ATP in our animal cell or in our plant cell. Chloroplasts, which are found in plant cells, not animal cells. Peroxisomes, vacuoles, lysosomes, and other vesicles. And each vesicle or each organelle is going to have the materials, the enzymes, the machinery it needs to do its functions. So if you are working at this superstore and you are working in the tire department, you're going to have the tools you need to be able to change someone's tires and to measure their tire pressure. Whereas if you're working in the toothpaste department, you're not going to have those same tools. You're going to have, the ability to stock toothpaste and to put the price stickers on your toothpaste. So each organelle has the different enzymes, which makes them different from other compartments, so that they're able to do the functions that they are specialized to do. Eukaryotic cells can exchange materials between those compartments, so things can move from the Golgi apparatus to the plasma membrane. Things can move from the nucleus to the mitochondria. Things can move between them. And a lot of this is going to involve a process called exocytosis, which is going out, exiting from one compartment, and endocytosis, which is pulling something in. This involves membrane fusion events. So a piece of membrane from one area of the cell exits, is pulled out, forms a little bubble that then travels to the next little area. So here you can see this little vesicle, these little bubbles traveling along, going to this next little organelle, and where that can then, this membrane can fuse with the new organelle, where it's going to be able to carry those components to different parts of the cell. Bacterial DNA is present as a circular molecule. It is still called a chromosome, although there are a few proteins associated with it. Eukaryotic DNA is a linear molecule. These are still called chromosomes, whether they're linear or circular. But in our eukaryotic DNA, we have a lot of proteins, those little spools of thread. The histones are those spools that the eukaryotic DNA is wrapped around. It helps protect that DNA from getting twisted up. Just like if you were a seamstress and you had a lot of thread, you're not going to have all your thread just loose on your table. You're going to wrap it around your spools. That's what histones are doing. They're helping organize and protect that DNA. Archaeal DNA is circular, although it is associated with proteins similar to the eukaryotic histones. And the packaging of bacteria. The DNA is longer than the cell itself, so it has to be folded and packed tightly, equivalent to packing about 60 feet of thread into one little thimble. Eukaryotic cells have about 1,000 times more DNA than bacteria have. That's a lot of DNA. And so we organize that DNA into chromosomes and pack those chromosomes up. So not only have we wrapped our DNA around those little spools, around those histones, we've also wrapped those histones organized around each other to create this nice folded up chromosomes. So when that cell divides, it's able to divide its chromosomes without breaking and getting things mixed up between each other. So we keep our cells nicely organized with their DNA. All right, so that's a pretty brief introduction to how our different types of cells are different. We're going to come back in our next lecture to talk about what is going on inside of our eukaryotic cell. We're mainly going to focus on eukaryotes this semester because it's really about, this class really is about mammals, so we're really going to talk about us. But it's nice to know that kind of background about other cell types out there. All right, let me know if you have any questions, and I will see you soon. Bye.